kind of asking the same basic questions. So today, just getting right into it. So obviously, as you all know, fish move around a lot. The ocean is a dynamic environment. It is always changing. And the way fish use that environment is all very dynamic and complex as well. Patterns of movement and habitat use are really dynamic and change constantly across spatial and temporal scales. So the first thing you have to do is kind of understand these patterns. You have to characterize them. That's really fundamental to understanding their ecology. But beyond that, you have to kind of dig a little deeper to get at the drivers. What's underlying those patterns of movement? What's actually causing the fish to be where they are when they are? That's going to be a mixture of a number of things. It's going to be a combination of abiotic factors, such as environmental conditions, habitat availability, all that kind of stuff. Biotic factors, so availability and distribution of prey, predators as well. And then also more inherent or innate variables, such as physiology. There's going to be physiological limitations or physiological optima and things like that. They're going to drive an animal's distribution. And all those things interact to form where a fish is and where it can be in its biogeography. And that in turn influences the ecology and its life history and overall fitness and evolution. And these are the kind of questions, after a long time trying to figure out what I'm actually interested in, kind of realizing I have questions I kind of consistently ask, was that what are these ecological, physiological, and environmental factors that really influence the distribution of marine species? And how does this vary across ontogeny and spatial and temporal scales? Now, it's one thing to do that in the kelp forest or in Elkhorn Slough. It's difficult everywhere, but for pelagic species, this is a particularly hard nut to crack. Pelagics are highly migratory. They move really far, really fast, and they're really hard to access. It's really hard to study them. They have a huge range. Difficult to study throughout their ontogeny from birth to death and across their range because they move so far. This is a track from a white shark tagged in South Africa that moved across the Indian Ocean in 9 months. So that's just one animal. When you're dealing with a species that moves across that kind of range, how do you study it? How do you know when the adults are moving across that area? And then the pups are only occurring in that area, and juveniles are only occurring in this area. The mating occurs over there. They're eating everywhere. So all these different things are interacting to influence the ecology and what that role of that animal is. And this is all critical to know if you're going to effectively understand and manage that species. So there's a lot of different ways people have kind of tried to approach this. And fisheries bycatch data or catch data is one way. The other one is satellite tagging. That's one thing my lab has been very active in. And so obviously, this is a great technique to use to understand the broad-scale movement patterns of a lot of different species and understand how they're using their environments and habitat use and environmental preferences. And we certainly learned a lot over the last 10 years with the development of satellite tags. This is a figure from Barbara Bloch's Tagging a Pacific Pelagic Synthesis paper, deployed over about 5,000 satellite tags on 23 species of 23 pelagic species. And they move all over. And so we've learned a huge amount about patterns of movement, habitat use, and behaviors of these animals. So it's a really powerful tool. But there's some really important limitations. So first of all, you can describe these patterns. They're going over here, and they're exhibiting these kind of diel vertical migrations or vertical distributional patterns. What does that mean? What are they actually doing there? You don't really know. You don't have a camera on them. You can't see what they're doing. Are they eating? Is it reproduction? Everyone always kind of says it's foraging or sex. That's the two things. So everyone says. But how do you really know? How do you get at it? The other thing is, how do you actually tag an animal, a species, through ontogeny? You have to be able to do this. You have to find them when they're born, tags on them, find them, but through all age classes. And when these animals live across ocean basins, that's almost impossible to do. It's also really expensive. These things are $2,000 to $5,000 a piece. So it takes a lot of resources, both to find the animals and tag them, but then to purchase the equipment, and then it's thousands of dollars just as satellite time to get the data back. So you really need to start bringing in complementary approaches, and this is kind of where I fell in the stable isotope analysis, some of these other approaches that I'm going to be talking about, which is how do you fill in these gaps? What other kind of approaches can you bring to bear to start to disentangle some of these ecological patterns you're seeing in terms of what they actually represent? So I think most people here are probably pretty familiar with stable isotope analysis, so I'm not going to dive into the details. But the basic is you are what you eat. The isotopic composition of a consumer is a reflection of its diet, and the isotopic composition of a prey resource is going to reflect where it lives and the food web it's a part of. So the way that you use these tools is completely dependent on a variety of different factors. One is the scale of baseline variability of stable isotopes. So these things can vary a huge amount across spatially and temporally, so it's typically called isoscapes. It also can vary a huge amount locally within a food web. So here's a California current, Southern California current pelagic food web. So depending on how much spatial variability there is in these patterns of isotopic variability and the pattern of movement of your consumer and its prey, that really is going to drive the kind of questions you can ask and how you interpret those data. So you can think of it in terms of like a dye experiment with different isotopic compositions having different colors. So here you have a pelagic food web. The primary producer sets the baseline isotopic composition. That's propagate up that food web. You can think of all the animals that are residential and live and feed within that food web being blue. Same thing for a coastal food web, an animal and consumer up here at the top that only lives and feeds within that's going to be red. An animal that moves between is going to be a mixture, so purple in this example. And so if you're thinking that spatially, and we've done some of this work for white sharks, you have white sharks that live over here and are spending time in the California current. They're going to look like California prey resources. Some that maybe spend a lot of time out of Hawaii, they're going to resemble offshore pelagic resources. And then you have some that maybe move back and forth, and they're going to represent a mixture of it. So the primary species I'm talking about today is the smaller cousin, but no less charismatic cousin of the white shark, the salmon shark. It's a highly migratory, wide-ranging endothermic species. They're known to elevate their body temperature up to 21 degrees C above ambient. They're highly opportunistic. They eat a lot of different things. We primarily focus on forage species. And they're endothermic. I'm going to be talking a lot about this. So endothermy is basically the ability to maintain a warm body. And there's a variety of anatomical and physiological specializations that are associated with this. One is the internalization of the red muscle. You can see here for a salmon shark, the red muscle masses are moved interiorly right next to the spinal column, as opposed to a lot of species that's exterior on the outside of the body. Then you have these REAT systems, these countercurrent heat exchangers, that basically what they do is the blood that's, the cold blood goes into the body towards the red muscle. Here's the red muscle mass. These are the REAT systems. They're these very dense capillary beds. So the incoming blood that's cold at ambient temperature dives in while the warm blood is going out, and as they pass, the outgoing blood dumps all of its heat on the incoming cold blood. And by doing that, it maintains the heat at the core of the body. And this is a very common engineering tool. It's used in a lot of things. These are some REAT systems behind the eyes of salmon sharks. It warms their eyes so they can actually see faster. And they also have to have very specialized cardiac systems, because one of the kind of basic conundrums you have is that the heart is always working at ambient temperature because of the way the circulation system is put together in fish. So if it's cold, the heart's going to be going slower. So it has to still be able to maintain this warm, hot body that's screaming for oxygen, and that heart has to be able to supply it. So they have a very specialized cardiac physiology that allows it to function at a high rate and high capacity across a range of temperatures. So a bit of background about salmon sharks is they are very wide-ranging as a species. They occur across the North Pacific. Now they have a really interesting pattern of sexual segregation. So as you look at the southern end of the range, they have an even sex ratio. But as you go further north, they become more and more skewed. And in the western North Pacific, you basically only find males, and over in our neck of the woods, you basically only see females. And we still, to this day, do not know when they come together. So that's a basic kind of question. So one of the biggest fish in the ocean, we don't know when they get together to make babies. And then you have size segregation. You only see the small ones at the southern extent of the range. You see the big ones everywhere, but you only get big ones up in the north. So the salmon sharks have been pretty well understood in the eastern North Pacific through the tagging of Pacific pelagics. We've tagged 150, 200 of them and learned a lot about them. So we know a lot about what they're doing. Well, I should say what females are doing. We've never tagged a male. But we know basically nothing about their early life history. We know nothing about what they're doing when they're young, really what they're doing in nursery areas, or even where they're popped. So what we do know, or what we think we know, is they're basically pups somewhere down in the subtropical gyre, and then they move to, or potentially born in, one of the two putative nursery areas that have been identified. The North Pacific Transition Zone, which has been identified based on bycatch of juvenile salmon sharks in the high seas Japanese fisheries. And then here in the California current. And the only reason we've identified that as a nursery area is because the babies wash ashore pretty frequently, as probably some of you know. So what are they doing? What are they doing in the California current? We don't know. No one sees them. The only time we see them is when they wash ashore. What are they doing in the transition zone? What are they doing up until they're big adults that live in Alaska? We have no idea about any of those. When do these shifts happen? It's some very basic questions that we've been trying to disentangle, and it's obviously been a difficult question to get at. But this is particularly important for a lot of these long-lived marine species like salmon sharks and lamnid sharks and sea turtles and things, where basically demographic elasticity analyses have indicated that it's a juvenile life history stage, it's survivorship that drives the overall health of the population. So it's really maintaining survival of those young animals that's what's most important. So I'm going to be talking kind of about 3 little vignettes today, kind of starting a little bit more zoomed in and kind of very broad scale. Basic questions first, kind of dive into what they're doing here in California current. What are they doing? And try to kind of, get at some hypotheses of why they might be stranding. Another is to kind of get at these potential ontogenetic shifts in habitat. Are there ontogenetic shifts in habitat we can identify? What may be driving them? And then start to get at this question of how does this endothermic physiology influence this distribution? So, what can we learn about how salmon sharks are using the California current? Like I said, the only reason we see them is when they're up close to the shore and they're basically dying or on their way to dying. And when people see these things stranding, it's pretty clear something's weird. Sometimes they'll stick their head out of the water like a seal, and they'll just wash up on the beach, and people pick them up and help them throw them back up, but they'll wash back up, and they're basically going to be dead one way or the other. So, basic question. They use the entire California current system from inshore to offshore. Do they primarily use neritic habitats? Do they use oceanic habitats? Are there latitudinal patterns to their distribution? What is the trophic ecology? What are they eating? Every baby salmon shark we've ever found stranded, the stomachs are empty, so we've never been able to get at any of that. So, how can we do this? Basically, the only thing we can do is use these dead sharks and learn as much as we can from them. So, we collected tissue samples to use stable isotope analysis to start to tease apart what we can about trophic ecology, habitat use, and then look at the actual patterns of stranding events to try to get at, okay, are there any patterns of where they are or not stranding? What did the ocean look like around the time that they were stranding? And to develop these hypotheses about why they may be stranding. So, what did we learn? So first of all, it's not a new occurrence. A lot of people, you'll sometimes read these stories always appear in the newspaper about salmon sharks or baby white sharks. They call them stranding, and people start waving their arms. Oh, it's contaminants in the water, this, that, and the other. But no, they've actually been stranding as far back as 1880 is the first record. And a nice little ichthyological callback was that guy was actually picked up by David Starr Jordan, the famous ichthyologist, first president of Stanford, who found a dead one up at Santa Cruz and sent it off to the Smithsonian. So if you ever want to go see the first salmon shark ever found, it's there in the Smithsonian. They're basically all young of the year, about 6 months on the average age, but up to 1.5 years or so, up to and all the way down to basically neonate. They strand throughout the year and all along the west coast. Here's a map showing the distribution of strand events. You see these kind of centers of distribution around central California and central and northern Oregon, which is probably more a result of where the people are. Again, these are all opportunistic records. Like every time I'd see a story in the newspapers, I'd record it and where it was if there's information. So this is not the best data, but it's what we have. And we're able to get our hands on 42 sharks. We collected liver samples, vertebra samples, and muscle samples. We didn't end up, I'm not going to talk any more about the muscle samples because you have issues with maternal influence, because basically when they're born, they look like mom, and it takes a long time for them to grow out of that. So these things, if you look at their muscle signature, muscle values, is going to be a mixture of what they've eaten, but also what mom was doing. So I'm only going to talk about liver and vertebra later on. So what can we learn about the patterns of distribution? So this is just by latitude and month of the year, California current sea surface, seawater, sea surface temperature climatology. And you can see some pretty nice patterns right off the bat. So their range extends north during this fall and summer and fall when the waters warm up. You basically don't see them stranding, or stranding, which we're interpreting means they probably aren't there, at water temperatures much below 11 or 12 degrees. So they're really almost all these stranding events occur in a pretty narrow temperature range between 12 and 16, but most of them between 14 and 16. And this actually matches really well with published data from the literature, from the North Pacific Transition Zone bycatch in the Japanese fleet. They say that in terms of where they only see juvenile salmon sharks basically between 13 and 16 degrees. So it matches really well with the other available data. So we think that this kind of shows basically where they are, and the records of strandings are matching where the sharks actually are. And if they're not stranding up here, they're probably not there. The other thing that became apparent was that they really are only stranding during the upwelling season. So this is the period of peak upwelling, and then the upwelling season by latitude. And they really don't strand outside that. And Of course, in the Southern California Current and Central California's pretty dynamic, upwelling can occur throughout the year. But that seemed to be a pretty interesting pattern that we started to dig into a little bit more. I'll get into a little more later. But in terms of stable isotope analysis, we were able to dig into the literature and go out and sample a lot of potential prey items and came up with 3 distinct potential prey clusters, which are basically oceanic mesopelagic species that basically live off the continental shelf pretty far offshore, ones that are on the outer continental shelf slope, maybe oceanic, and then these prey that are going to be kind of represent more inshore coastal neritic prey resources. And this is basically how they fall out in carbon and nitrogen isospace. So we can take those data and throw them into a Bayesian isotope mixing model, which basically takes into account error in the prey sources, error in the consumer sources, error in a variety of different factors, and basically gives you an estimate, a quantitative estimate of how important, how much of those different prey sources those things would have to eat to have the isotopic composition they do. So if you look at the vertebrae, now the vertebrae that we sampled were everything outside the birthmark, so basically everything that shark didn't eat from birth to death. So cumulative lifetime isotopic kind of trophic signal. This is what we found. They're primarily feeding in these offshore shelf slope and then oceanic resources. So can't really say what they're exactly eating, but what they're eating is going to be more indicative of stuff that's out there, which makes sense given we never see them really. Then if you look at the liver is very metabolically active and does represent recent diet over the last weeks or month or so. You see a subtle shift where basically you see the biggest change, you see a drop in the shelf slope. Oceanic stays about the same, but you see an increase from basically nothing to 10 to 15% from this kind of coastal neritic signal, which we interpret to mean that basically right before death, these little sharks kind of moved in potentially to more coastal habitats and started to feed and pick up this coastal prey signature. So what we did then was we took every strand event and said, okay, what did the ocean look like in the previous 2 weeks prior to stranding? Because 2 weeks ends up being an important time frame because these animals all die of this bacterial infection I'll talk about a bit more later, and that's about the longest they could survive with the infection. So what did the ocean look like in that area? We looked at a 2 by 3 degree area. We looked at a variety of different oceanographic parameters, sea surface temperature, chlorophyll, but the main ones we were interested in was temperature and upwelling. And we used meridional winds as a direct proxy for upwelling. And really the only thing that came out as being important and actually being very significant was sea surface temperature. And we had 2 sea surface temperature proxies or parameters. One is the average sea surface temperature in that box, and then that minimum. So it was the coldest sea surface temperature that occurred in that box during the previous 2 weeks. And what we found was that basically the probability of stranding is highest when the average sea surface temperature was above 14 degrees, which is again showing you kind of that preferred habitat that they seem to be following. But they really only strand when you also, that's associated with these episodic periods of very intense or drops of temperature, where sea surface temperature drops 3, 4, 5 degrees down to between 8 and 10 degrees. So the idea is that they're there in this nice kind of toasty warm water, and maybe there's some minimal upwelling event that occurs and pulls up this cold, deep water and gets them. And that's going to be associated with these increased probability of stranding. So we kind of put this all together to come up with this hypothesis of why they're stranding. That's still what it is, our best hypothesis, but why they strand. So almost every shark that's actually been studied that's stranded, when they look and do a full necropsy, they've had these massive meningoencephalitis infections. They've got lesions all over the brain, sometimes it spreads to the liver, and it's just all over. And it's caused by this carnobacterium, which is well known from the aquaculture literature and a lot of things where it's it's a common part of the floral community of a lot of temperate and of fishes, especially well known in salmon, in aquaculture salmon. And so what happens is these aquaculture salmon, for example, when they're handled, when they spawn, they get stressed out. This bacteria that's always there becomes pathogenic and kills them. So what we're thinking is that maybe this bacteria is common because they find it, they've identified it in the salmon shark, maybe like a lot of other animals, it's on every animal, and some environmental stressor stresses the animal out, compromises the immune system, and leads to this, this infection. So basically what we kind of put together with all of it is, okay, so juvenile salmon sharks are using the offshore waters of the California Current. That's their primary habitat, and that's really why we never see them. But then, based on the isotopic analysis, suggests these animals move into coastal nearshore waters, maybe following prey resources start to feed, they start to incorporate that coastal signature. And if they're there at the wrong time, they get stuck during the upwelling season with these very cold, intense upwelling events. And that, for these small endothermic sharks, may be a thermal stressor It just knocks them off, stresses them out, they get the infection, they either die outright or they get the infection and end up washing up on the beach. It's a nice story. I don't know if it's true, but it's our best guess. So that's kind of what they're doing here, we think, right? That's our best shot so far. But okay, there's another nursery area. Which one's more important? If you're going to protect one versus the other, how do you kind of prioritize those efforts? Just describing those patterns is a basic question. And then when do these ontogenetic shifts in habitat occur? And so how can we actually start teasing this apart? And so what we did was we sectioned the vertebrae. Here at Moss Landing, used the micromill just right down the hall. And so as you all are familiar, salmon sharks and sharks in general, they lay down growth bands with basically year by year, they lay down these light and dark band. Salmon shark's been validated, so we know that it's an annual growth band. So basically what we did was we used the micromill to collect all the tissue in each growth band, and we analyze that for stable isotope analysis. So that's going to tell you basically an integrated trophic and habitat use signal for everything that shark did and ate and went during that year of its life. And so then we can make a life history profile of what that animal did from birth to death. These were all sampled from adult animals caught up in the sport fishery up in Prince William Sound, Alaska. And so this is the basic data. So you see a nice clear ontogenic shift. Here's carbon, here's nitrogen by age. U is in utero. We took tissue from inside the birthmark, so it basically represents all the growth and development that happened in utero. So you can see that basically in utero they look basically kind of like mom, which makes sense because functionally they're eating mom when they're in utero. Then you see nice low pattern early in life and pretty high carbon nitrogen later in life. So when you translate that in terms of the broader eastern North Pacific isoscapes, what we did was we found data for about 50 to 60 different prey species across the North Pacific from 40 or 50 different studies. We came up with average prey sources for these different boxes, which are based on Longhurst biogeographical provinces. Note there is the North Pacific transition zone here, which we kind of ignored because we didn't really have good data, so we basically lumped everything in there together. But so basically, all that early life history is down here, and as they grow, they move up this way. And so the young animals are all down here, the old animals are up here. So this is going to be more indicative of these oligotrophic, offshore, blue water habitats. This is going to be more indicative of highly productive coastal neritic habitats generally. So one thing we noticed was that if you look at year 0, so the first year of life, there's a pretty broad distribution in carbon and nitrogen. There's a lot of variability. And we interpreted that as maybe representing differences in nursery use. So California current versus the North Pacific transition zone. So we went back to our vertebrae collected from these little dead guys, used them to, as training data and discriminant analysis to characterize what the California current nursery looks like. All those white triangles is all the juveniles that were stranded. And then we used a subset of the data that we're identifying as the transition zone signal, which is basically going to be these couple of years offshore where they're low and consistent, and when you look at the prey values, they're more indicative of that kind of habitat. And then we basically characterized all the year 0 animals into belonging to the California current nursery or the North Pacific transition zone nursery. And you can see the results here, and basically 30% of the animals, again, these are adult animals we caught that were caught up in Alaska. Looking back in time, 30% of them used the California current as a nursery area. The other 70% used the transition zone as a nursery area. And then what was kind of interesting was when you look at year 1, all the animals, even the ones that used the California current, moved offshore. They moved out to the transition zone. So they didn't just stay in the California current and live here and grow up and then move north. They were born here, lived here for some period of time during the first year of life, and then they skedaddled offshore. They stay out there in the transition zone, if you just look at this consistent pattern for several years. Then they transition up into this more adult type signal. They start to mature around 7 or 8, 6 to 9. So, in adults, they're pretty consistent carbon nitrogen values across the different age classes. So then we were interested in, okay, what can we learn about when these shifts happen? You can kind of see it with your eye, but you want a quantitative way to start taking it apart. So we used discriminant analysis again. We used the last several year classes to characterize adult habitat, and we used the first several, years 1, 2, and 3 to characterize the nursery habitat. And then we basically assigned each data point for each year class into how many belong to whether it belongs to adult or juvenile habitat, and got a proportion that are using one or the other. And we fit a logistic regression to it. You can see here what that is. And what we found was that basically the shift happened starting around age 6. That's when they make the shift from juvenile to adult habitat. And that corresponds with the age of maturity. They start maturing at age 6, and they mature about through age 9. The youngest shark ever caught and aged in Alaska was 5 years old. The average age of every shark caught and aged in Alaska is 11. This histogram is just an estimated age of every shark we've caught and tagged as part of the tagging of Pacific pelagics up in Alaska. So all this really kind of lines up pretty well and suggests that this timing of the shift matches what is actually going on. So all the data we could throw at this supported our isotopic analysis. So then we actually looked at how does the isotopic niche shift through ontogeny. And this is going to be basically a metric of the it's a rough proxy for the ecological niche of the animal. So what are all the resources and habitats these animals are using? So a more dispersed area, a bigger ellipse is going to represent a broader niche. Smaller niche is going to represent a more focused, specialized consumer. And so what we saw was here, you can look at the ellipse area, and that's basically these colored areas by age. They basically get smaller with age. And so it's basically the indication of that is that they get older, they become more specialized in potential habitats or prey, which was kind of surprising to us given these things migrate all over. But actually, when we started looking at the data, it actually ended up kind of making sense, which I'll talk about in a second. And part of that is this salmon story, which I'll get to. So the good thing was that we actually had a lot of the satellite tag data we could actually bring to bear directly to this question. So we had 45 year-long tracks of salmon sharks. So we could actually say, what % of the year were these sharks generally spending on these different boxes that our isoscape was representing? And we could use that as a prior and incorporate that into the Bayesian mixing model. So basically it allows us to incorporate what we know about their distribution into the mixing model. And this is what we found, basically in terms of time spent in different areas. These sharks really spent most of the time in that Alaska box, the coastal waters of Alaska and the subarctic gyre, followed by California, and then not much time really in the pelagic environment. If you look back, there's a lot of points along the neritic area, but especially really the density is up here in Prince William Sound. Kodiak Island is really kind of where they're focused. But so anyway, we're able to actually incorporate that. The great thing about these Bayesian isotope mixing models, you can actually incorporate these different kinds of information as prior information that helps inform the mixing model results. And this is what we found. So again, these are what these boxes mean. You know, Alaska, California, I'm calling them neritic boxes, basically neritic ecoregions, because they have shelf habitats. And then there's the two oceanic ones, the subarctic gyre, transition zone, and the subtropical gyre. And this is the proportional contribution through ontogeny of those different ecoregions. So quickly what you can see is that, all right, early in life, there's very little contribution from neritic habitats. You can see a little bit potentially in that first year of life, which is going to be those California current sharks. But then you see a shift here, which corresponds pretty well with what we're seeing around 6 to 9, to increase use of these neritic habitats, in particularly Alaska. So this is going to be, the dots are going to be the median, interquartile range, and then the 95% credible intervals. So this is basically the two mixing models we ran. One ran one uninformed without using the tag data, so basically a pure trophic tracer. If you're assuming that every shark is a generalist and it's eating everything equally without any knowledge of where it's spending its time or anything, that's what this uninformed mixing model is telling you, versus the informed one, which says we know something about how much time they're spending in these different ecoregions. So that's the solid line here, and the dotted line is the uninformed. So it ended up being kind of interesting looking at the differences of this. And basically, there's no difference in here. We only did it for ages 6 on, because that's when the animals are up there, so there's no point incorporating tag data for adults in terms of what the juveniles are doing. But so what's interesting then is, like, well, thinking back, why do you have this difference? You know, the uninformed model is saying there's a much higher contribution of pelagic prey resources than we're seeing from the informed model. So what you'll see here is 4 pictures of salmon sharks up in Prince William Sound eating salmon, Pacific salmon, so pink salmon, sockeye salmon, things like that. What you'll also notice is there's land everywhere, right? These salmon live and grow up in the middle of the ocean. So what these salmon represent is they've integrated resources from the high seas and brought it to the mouths of rivers. And these sharks show up at these river mouths when the salmon are forming up to go run up the rivers for spawning. The sharks show up at the same time and just gorge on them. So this kind of represents a trophic subsidy. These animals don't have to go anywhere, and they're integrating prey resources from across the entire North Pacific, because they basically sit there with their mouth open, the salmon just swim by and they eat them. So it helps us kind of disentangle the provenance of the resources they're eating, so where the prey are coming from versus where the sharks are actually feeding and spending time, which ended up being kind of an interesting little twist. So what did we learn? So the basic kind of conceptual perspective we've developed is, okay, this is a 4-year track of a female salmon shark doing the same loop over and over and over. We think that she goes down here to give birth. So the females are born somewhere down here. Again, we still don't really know. They move to one of the pupping areas, California Current, or the North Pacific Transition Zone. They move out, the ones that don't die, move offshore, and then they stay there for years 1 to 5 or 6 or so. And this is when they're most vulnerable to the high seas fishing pressure. There used to be a huge amount of bycatch in the high seas. They would catch up to, it's estimated, as much as 100,000 juvenile salmon sharks a year. That stopped in the early 90s when they put the curtailment on the high seas drift gillnet fisheries. But so then that kind of leads to this question of why did they use the North Pacific Transition Zone? Why not stay in the California Current? The California Current is so productive, so much food. Why would you go out to the middle of the ocean and live out there for all of your early years for the most part? So North Pacific Transition Zone is this band that goes across the North Pacific. It's basically this chlorophyll front where you can think of it as going from the kind of green northerly cold waters to the blue clear, tropical waters. It's very tightly coupled to the 18-degree isotherm. It migrates seasonally about 1,000 miles latitudinally. And it's a very well-known, here as you can see the chlorophyll front there. That's going to be the equator, so it's all the blue, clear ocean water. It's a very well-known migratory corridor and foraging habitat for a variety of pelagic predators. Blue sharks, dolphins, albatrosses, sea turtles, all these different things. They migrate across it, they forage across it. It's, in terms of being a pelagic environment, it's probably one of the most productive ones you can find. So, why use the North Pacific Transition Zone as a nursery area? One hypothesis is there's fewer predators. So indeed, the biggest predator of small sharks is bigger sharks. That's pretty much the only thing they'll eat. Basically, in the north of the transition zone, there's really nothing there except for bigger salmon sharks. There's no evidence of cannibalism, but you never know. There really aren't much. There's some blue sharks that kind of scatter up there, but really not much north of the transition zone. But there's a lot to the south. There's blue sharks, there's Makos, there's oceanic whitetips, there's white sharks. In the coastal waters of California, there's all that. There's seven gills. There's a lot of different things. So basically, everywhere else, there's a lot of predators. And this is actually of concern for even a big animal like a salmon shark. This is a salmon shark we tagged up in Alaska in 2003. This is its track. It moved down here very happily, and this is basically a reconstructed water column. So it's diving from the surface down to 100 meters or so. This is color-coded by water temperature. They hit warm water. They don't like the real warm water, so they exhibit submergence behavior, so it stops going to the surface. Gets to about here, then bang, something happens. So if you see this is something keeps diving from the surface down to about 350 meters at a pretty much constant 26, 27 degrees. Any ideas? That's the internal body temperature of a white shark. So that shark was swimming down, and a bigger, badder predator came along and gulped him right up. And what's actually interesting with the top data set is we're actually seeing a whole lot of evidence of this. So you kind of, wow, it's such a big, open, empty ocean. We've had swordfish, marlin, blue sharks, everything you put a tag on ends up getting eaten by one of these things out there eventually. It's pretty amazing when you think about it. But, so predation is actually an issue. And then so that's basically the white shark diving up and down. Actually, it's kind of interesting. You can see here, the day after it ate it, just stayed up at the surface. You can kind of think it's basking. It's like, oh, I can't believe I ate the whole thing. It's not swimming, up and down. It takes a lot of energy, actually, to digest and move that bulk of tissue through the GI tract. So, okay, fewer predators, maybe, makes sense. What about prey? Again, like I mentioned, it's well known that actually this is very dynamic. There's lots of frontal systems, lots of mixing, lots of eddies moving through. It's actually a very productive area for pelagic habitat, and a lot of animals focus on this area. So if you're going to be there, that's going to be where there's probably going to be a pretty high density of prey. But then also importantly is going to be this thermal habitat issue. So these sharks, if they like this 14 to 16 degree temperature range, they can just migrate seasonally with this front and be always at the constant temperature they like, not worry about upwelling or any kind of variability. There's going to be fewer predators, and there's going to be ample food, at least for pelagic habitats. And maybe the California current is simply too variable. Too many predators, thermally too dynamic. So it may be this trade-off between, okay, we don't want to get eaten. We want to have as much food as we can, given the context that we don't want to get eaten. And we also don't want to strand because it gets too cold. So it may be this kind of optimal trade-off between all these different factors. So then, between ages 6 to 9, they mature, and the big ones start moving and utilizing neritic habitats. So why make this shift at maturity? And our one hypothesis is that this is increased energetic requirements associated with reproduction, or potentially change in endothermic capacity. The bigger you are, the better able you are to maintain a warm body and utilize these cold waters. But so this change of life history requirements is going to probably be pretty important. So being a pregnant female salmon shark, it takes a huge amount of energy to provision your young. These are ovophagous animals, so basically they pump out nutritive ova, so eggs that basically those baby sharks just sit there and eat. It takes a huge amount of energy. Gestation is about 2 years or so. They give birth to 4 or 5 big pups. It takes a lot of energy. So it may be that they have to move to these coastal, highly productive environments to get enough food to make the babies. Then maybe it's this issue potentially associated with endothermy. Maybe they just aren't big enough to move up to Alaska until they're that size. They may not be able to maintain a warm body and function physiologically at those high latitudes. But also another theory is that it's to reduce competition with the younger sharks. It's kind of hard to test that, but that's another idea. Then there's this idea that the adults become specialized. And indeed, we know that when they're up there, they do seem to focus and exhibit these very consistent behaviors. Here's 3 different sharks showing, this is a shark that spent 3 years just going back and forth between Kodiak and Prince William Sound. This is one that we tagged up there, came down, spent 3 years off here, California. This is one that did that loop 3 years like that. So you see these animals that exhibit the same behavior over and over and over again. And when they're up there, they're probably eating 1 or 2 things, salmon or pollock or squid or something like that. So this decrease in niche breadth actually kind of makes sense, even though some of these animals are moving across pretty broad distances. So then, we've been talking a lot about this endothermy, and then what we kind of said, well, let's step back. Does this make any sense? Everyone who works on lamnid sharks or endothermic tuna, bluefin tuna, we always talk about, the bigger they get, they can go into colder water, but no one's ever really tested this or got at this question. So we were curious about diving into that. So really quick, again, talked a little bit about endothermy, but lamnid sharks are one of the few. So lamnid sharks are going to be the white shark, the salmon shark, the poor beagle, the short fin mako, the long fin mako. But they're one of the only species of fish that have taxa fish that have evolved endothermy, with the tuna also doing it, and then some scombrids generally having some different kinds of endothermy, different flavor, like cranial endothermy and things like that. But being endothermic is well known, and everyone always kind of weighs around about how great it is. It really kind of makes you be able to swim faster, have a higher metabolic rate, higher aerobic performance, broader thermal niche, broader geographic and vertical distribution, all these different things, longer migratory behaviors. But there may be some limitations. So really quick, I finally did this. I was like, I have to get together a schematic of how the REITs really work in case someone doesn't understand it. But so this is going to be the red muscle. The red muscle here, again, is here, this internalized muscle mass. That's which generates the bulk of the heat of these animals. So the final outcome of all energy, everything you eat, is heat production if it isn't actually incorporated into muscular growth or something like that. So every time these animals are moving, they only use the red muscle to swim. They're always using it. They're ram ventilators. They're constantly swimming, so they're constantly generating heat. These generating the heat, the warm blood leaves. They go through these dense capillary beds, so the warm blood's going out. As they go in, they pass the incoming cold blood, pass off their heat. They hit the heart, and the heart is outside the REIT system, therefore it's function at ambient temperature. So you have this whole mass in terms of the viscera, because these sharks also have REIT systems in their hepatic REITs, in their kidneys, in their livers, in their eyes and brains. So these have REITs all over the place. So this animal is a very high performance predator, high metabolic rate, needs a lot of oxygen. So this heart has to be able to provide that body with enough oxygen. So they have the specialized circa-2 protein and these ryanodine receptors, which increase calcium cycling in the heart, which allow it to basically pump a lot faster than is possible in anything else, even at very cold temperatures. So and they also have huge hearts. Then they go through the gills. The gills, basically, any remaining heat will instantly go to the environment because thermal diffusion occurs 10 times faster than molecular diffusion. So it's always going to lose the heat before you get the oxygen. That and there's only one cell thick between the respiratory epithelium is only one cell thick, so basically you lose all heat at the gills all the time. So, and famously, Frank Carey, who's a famous biologist from Woods Hole, who's did a lot of really foundational work, fundamental work on tuna and sharks, he ranked lamnid sharks in terms of their ability to be warm. And he put salmon sharks at the top of the pile. Again, they can be very warm, and we've estimated the REITs to retain about 99% of their metabolically generated heat. White sharks are a bit lower. Their REITs aren't nearly as effective, and they maintain body temperatures up to 10 to 12 degrees above ambient. And the short fin makos have the lowest degree, but they still have pretty effective REITs. And here you can see a different cross-sectional, so the salmon sharks, white sharks, and makos. You can see how the red muscle mass is generally a little bit more evenly distributed and further closer to the periphery. So lots of lots of benefits, but there's also some pretty significant potential limitations that no one really thinks about, although we talk about them all the time in terms of thermal inertia and being small. And this is work from Diego Bernal, and basically it's showing you, based on temperature and tailpipe frequency and relative power, how well this red muscle functions across different temperatures. And what's important is if this muscle temperature, red muscle, drops below 20 degrees, it basically stops being able to perform work. So if this red muscle gets cold, this shark can't swim, and then you're done. So there's this lower thermal threshold these animals can't cross if they want to survive and function. And there's evidence for this in salmon sharks and for Makos, for yellowfin and bluefin tuna, and a variety of other species. No one's ever looked at white sharks, but they're in between these, so it's going to be the same for them. So clearly this is going to change a lot through ontogeny. This is a neonatal white shark and a big white shark. That shark's going to have a lot harder time staying warm in a cold ocean than this big shark is. So these thermal limitations may be particularly important during the early life history. And indeed, we have plenty of evidence, and some of the stuff I've talked about with salmon sharks, the stranding and things like that. But juvenile white sharks are known to skedaddle. They leave the Southern California bight when water temperatures drop below about 14 or 15. They go south to Baja. White sharks don't occur north of preconception until they reach a certain size, and people have kind of suggested that's related to endothermic capacity. So we wanted to see if we could come up with a way to get at this question of, can we get at a kind of a guesstimate of what's the coldest water temperature a shark can be in and still be a warm shark? And how does that change across ontogeny? And then how may all these things kind of interact to influence habitat availability and distribution, and how that changes through ontogeny? And again, when I'm talking about thermal habitat, I'm talking about a habitat envelope, kind of a range of temperatures they can be between, not like preferred temperatures where they are. So what we did was, myself and Mark Denny, who was fundamental and really helpful for this down at Hopkins Marine Station, we created a biophysical heat balance model based on first principles of physiology and physics, based on Newton's law of cooling, to basically come with this estimate of how cold can they go. And it takes into account a variety of different parameters. This is a very simplified version of the equation that I'll show you later. But basically what this does is Newton's law of cooling describes the rate of change of body temperature given a certain thermal gradient between the interior and the exterior. And you can model a bunch of different things. And if anyone's interested, this is kind of the Bible. So this is a great paper, and we learned a lot and used it. It was really fundamental. So this heat balance model has two primary components: heat production and heat loss. So heat production, we're talking about two different sources. It's going to be basically the heat produced through the standard metabolic rate, or basically the cost of the heat you're produced simply by being alive, and the heat produced by swimming, so the red muscle activity. And then heat loss is going to occur across two mechanisms: heat loss across the body wall, just convection through the tissue, and then heat loss across the gills, which is going to be mediated by the Reed efficiency. And I forgot to do that. So going a little bit more into detail on this, because this is kind of a little bit newer, and I always want more input if anyone has it. So it's well known that metabolic rate scales with size. Mass-specific metabolic rate goes down the bigger you get. So there's some good scaling relationships for laminid sharks that are published by Manny Escura down at the aquarium and others, and Sepulveda. And so we're able to use some published literature to actually scale what the standard metabolic rate is with the size of the animal and how much heat that produces. And then to get at swimming speed, the other issue is the speed with which you swim through the water is going to vary depending on how big you are. So the smaller you are, proportionally, you swim a lot faster than a bigger animal does. So fortuitously, this paper by Yuki Watanabe came out right when I was doing this, where he created a scaling relationship for endothermic fish, where he provided a relationship where you can scale swimming speed by body mass. It was perfect timing. It was wonderful. It made me so happy. So I was able to use that to scale how fast a fish should swim at a given size. And then we used kind of a classic biomechanical model to estimate basically how much energy does it take and how much heat is produced to push an animal of a given size and shape through seawater. And that's how we get at that swimming speed issue, because we really want to be able to play with swimming speed to see how important it was in terms of driving these patterns. So estimating heat loss. So again, two primary components: heat loss across the body wall, heat loss across the gills. So the primary thing is this thermal rate coefficient, which is simply a kind of an integrated variable that takes into account all the different ways in which heat moves across the body wall. But the way we're using this is basically just heat across the body wall. So it's just how a chunk of meat warming and cooling, how quickly does that happen given a certain thermal gradient. So we did an experiment, one of the more morbid experiments. We took some dead laminate sharks, we thawed them, we stuck temperature sensors in them, we warmed them up and cooled them off, and we had sensors outside so we could get that K value across different size ranges. So again, this reflects a reed efficiency of 100%. So it's the same as saying the reed is retaining all the heat. Nothing is being lost through the gills. That allowed us, this is some of the raw data just showing you how temperature changes. It really allows us to scale this K value by size and scale that accordingly. Then we had to incorporate the reed efficiency. So if you know it's retaining a certain amount of heat that's produced by the gills, there's some published data that kind of indicate that, well, you can see here that here's the heat exchanger efficiency by the relative to the predicted thermal excess, so how warm you are relative to the environment, that reed efficiency is really important. And very small changes in reed efficiency, when they're very effective, so when you're talking about 96, 97%, result in a huge change in potential thermal excess. So if you have a 99% versus 96% reed efficiency, it doesn't sound like a lot, but that's going to make a difference between potential thermal excess of like 14 and 6 or something like that. So it's really important. So we do all this stuff and do lots of great algebra and throw all these parameters. This is the final model. It doesn't really matter. Don't pay attention to it. It takes into account a lot of different things that we all scale where appropriate. But what this does is this final monstrosity lets us estimate what the minimum water temperature is a shark can reside in, stay in, when it's at thermal equilibrium with the environment. So heat production equals heat loss. So this is the absolute coldest it can be and maintain a certain minimum body temperature, which we're setting at 18 degrees based on how warm or how red muscle interact or reflect the response to cold temperatures. So after it drops below 18 or so, it stops working. So that's where we're setting that lower limit. You can change it to whoever you want, just changes the final result up, 1 or down. It's just a linear relationship, so it doesn't make a big difference. Then we wanted to say, okay, how does that actually reflect what we see in the real world? So we only are talking about sea surface temperature. And we wanted to find all the data we could find. So we looked at bycatch data, drift and inshore gill nets, the juvenile shark cruises, the nymphs perform, stranding events, and all the satellite tag data we could get on. And we looked at all the sea surface temperatures associated with each one of those records. And we're only talking with sea surface temperature because, again, that's the warmest part of the water column. That's going to be, an animal, I'm not saying it can't dive and go to colder waters, but it can't stay there. So it has to be warm enough where it can dive down, come back, and re-warm to maintain that minimum temperature. So just sea surface temperature, not total vertical niche or thermal habitat or anything like that. So how does it actually work? So this is the actual final outcome. That's a little light, sorry. That's minimum predicted temperature for this Mako, white, and salmon shark by age, out to age 20. So basically what you can see is that during the early life history, all the animals are pretty restricted, can't be in very cold water, and pretty quickly as they grow, that thermal envelope changes and increases. They're able to use colder and colder water. Now salmon sharks didn't really get into it, but we took into account body shape. You look at this is a little short, stubby guy. These are long and thin. They have very different body shapes, which is going to influence your surface area to volume ratio and how well you can retain that heat also. So body shape and reed efficiency are hugely important in this. But again, they're most thermally constrained at the small sizes. This is the same thing, just showing the different reed efficiencies. This top one is 100%, 99%, 97, 98, or I'm sorry, 100%, 99, 98, 97, 96. So this is you can see how important these very small changes in reed efficiency are. So if you're just losing heat across the body wall, and this is for a white shark, salmon shark, Mako, and a white shark, that difference in potential thermal excess is huge. So we don't really know reed efficiency. We're kind of guessing based on the literature and other things, but it's important. But again, if you look at the early life history, it doesn't matter. They're still going to be really thermally constrained because they're so small. And we did a lot of sensitivity analyses to indicate which parameters are really driving this pattern and driving the results. But really what fell out is that swimming speed is hugely important. So here's age. You can use size as well, and basically swimming speed from 0 to 2 body lengths per second. And it's color-coded by minimum water temperature it can be in. So if the shark was swimming at the same speed, relative speed across ontogeny, you just think of like drawing a line across there, and that's going to be what its minimum temperature is. But this is the predicted speed, so that's what we're actually showing. But you can see what's actually kind of, again, a small shark can change its swimming speed a lot, and it doesn't make a big influence on how much heat it produces and what the minimum water temperature can be in. But a big shark doesn't have to change its swimming speed very much, but it's so big, it retains heat so well, it produces a lot more heat and can be in much colder water. So swimming speed is behavioral, right? So they can change that. They can decide to swim faster, make more heat, or not. That's going to have an energetic cost. They're going to have to feed the body, but it's behavioral, so it's not a fixed parameter. So it's something that's one of the reasons we included swimming speed in this, so you could figure out how much they could actually just decide to change this. So here's how well it works. All right, so this is all the data we have from Mako. About 10,000 catch records, about 3,000 days from satellite tag data. This is basically the proportion of records at a given sea surface temperature by mass, and on the top is age. So the model output is like this. So this red is going to be what we think is actually the actual read efficiency, 97%. Actually does a pretty good job. You actually see very few records of sharks occurring at temperatures below that. And actually, again, if you look at these early life history stages, you can see that they indeed are more restricted. This is for white sharks. So this is about 55 catch records and about 13,000 days of data from 106 tags. And there's the results. So 97% is what we're estimating. Again, does a really good job. There's almost no data that actually fall below that minimum. And actually, you would not expect it to, because that means it's going to be a little more work, right? You'd expect them to leave before that. But that's really encouraging. Again, if you look at this early life history, it's most constrained. And this is indeed 15 degrees is about when they expect. Once the temperature drops below that, that's when they kind of leave the area. This is for salmon sharks. We have a little bit of data early on, not a lot in between. This is all the tagging data. And that's what we see. So again, pretty good job. If anything, the model underestimates how much heat they can produce and how well they can retain it once they're big. And that, I'm not sure why that is. It could be any number of things. They're eating more, they're swimming faster, their reefs are slightly more efficient. A lot of the big bluefin tuna, they're estimated 99% plus read efficiency. And again, thinking back to this, that matches pretty well with where we're actually seeing them in the real world and where you stop seeing them. So it actually does a pretty good job. I have a beautiful movie that doesn't work, I don't think. I'll try it. Nope. So anyway, what that just does is showing you how habitat availability changes through size, and it was 20 years of data. So only big sharks, red sharks, or 20-year-old sharks can be up there. Small ones can be down here. But once you get that kind of information, you can start looking at time series approaches, like how much of habitat is available in the California current. This is back from 1982 to 2014. And this is color-coded by age. So 0, basically, the purple is where a juvenile can be. You can see the amount of habitat changes hugely over the seasonal cycle. And for Makos, they're pretty influenced all the way up through age 20. White sharks get so big that by the time they're at age 10 or so, they don't really care. Then you can look at how these patterns are influenced by broader oceanographic regimes, El Nino, the ENSO cycle, or the PDO. This is the Pacific Decadal Oscillation Index. Positive is, the warm cycles, negative are the cooler cycles. And the lines are different ages. So again, when you're a juvenile animal, the nursery habitat, the amount of nursery habitat available to you is potentially changing a lot year to year. But by the time you're a big animal, you're going to be much less susceptible. So really quick, what are the implications of this stuff? So this California current's a shared nursery area for the 3 landed sharks. So what's going to happen in the future as a result of climate change and other things? So kind of counterintuitively, upwelling is projected to increase in certain climate change scenarios, potentially decreasing the sea surface temperature. The phenology of the upwelling season, the location of the upwelling centers are going to shift, all these different things potentially. We don't really know yet. They're still working on all the climate change scenarios for the eastern boundary currents, because the global models actually really suck for eastern boundary currents. And we're waiting on those because we'll actually feed that stuff into this to get an idea. But what's really important is that we have there's been some work to kind of estimate by Elliot Hazen and Sal and others, Stephen Bograd, how is the distribution of other pelagic predators going to shift in the face of climate change in the future? And landed sharks, the white sharks, the Mako, the salmon, and the blue sharks have been predicted to be basically losers in the future. They're going to be losing habitat overall. And so what we've shown is that, well, given what we know, it's that early life history stage, which is really important. This is all for adult habitat. We don't know what's going on in juvenile habitat. And indeed, it may be what's going on in this juvenile habitat, which is going to be more important in broad scheme of things. So whether that juvenile habitat goes up or down may be what actually mediates the overall impact that climate change is going to happen on these pelagic predators. In addition, something else that's kind of, I think that's fundamental, but we've never really kind of thought about before, was that these animals, what is the implication of endothermy on pupping? So these animals, when they're in utero, are at a constant 25, 26 degrees C. They're thermally acclimated for 2 years at one temperature. Physiologically, that's really important. It influences cell membranes, enzymatic activity, all the different physiological parameters are going to be hugely sensitive to any change in water temperature. So all lamnid sharks are believed to go on these large-scale putative puppy migrations, but no one really ever says why. Here's a salmon shark, here's a white shark, here's a Mako shark. Porbeagles are basically salmon sharks in every other ocean basin. They go to the Sargasso Sea. Why? And I think it's because there's going to be cold shock. If you're a pup, you're thermally acclimated to a constant temperature. You can't be born anywhere except for somewhere that's going to be, the water temperature is going to be close to your mom's body temperature. And because when these pups are coming out, they're not generating their heat. They're not swimming. You know, they're going to have to get their legs before they can start producing their own heat and swimming and doing their own things. So these temperature effects may underline evolutionarily why lamnid sharks go to warm water potentially to give birth. And that is something we're seeing, and we saw it this year, right? So people were paying attention. There were juvenile white sharks for the first time up off Santa Cruz. Last year there was a neonatal size one right in Monterey Harbor swimming around when these water temperatures were huge, were really warm. It's the first time in record anyone has seen this. So I think that's totally a thermal effect of this thermal limitation, an increase in this potential nursery habitat. So in summary, yeah, it's like to get these pelagic species that are difficult to study, you really have to take an all-of-the-above approach. Satellite tagging, isotopes, everything you can, physiology, to really get at it. And by combining all these different approaches, you can actually not just characterize the patterns, but potentially get at some of these drivers. And that's it. And obviously, that's a lot of work from a lot of people. All my collaborators for these projects are up here. But yeah, anyone have any questions? Happy to take them.
How long can they be in that cold water?
Right. In other words, are the ones getting caught in here the ones that actually came in, or are there many more coming in and some are able to swim out?
I think because I think that's totally accurate. You have an area of upwelling, it comes up, thump, and if you're in the middle, you may be screwed. If you're on the outside, you can swim out fast enough where your temperature doesn't drop. But yes, I mean that's something that the next step in the model is building like a higher resolution to get it. How quickly the body temperature dropping, thermal line, how long you stay for your boat. It's that kind of short-term, high resolution. Yeah, exactly. Yeah, it's absolutely.
I have a question that was said in one statement that you saw untagged salmon shark, white shark. And you said that predation was important. So I started thinking, well, how important is predation versus prey availability? And of course, my answer would be off the top of my head that it's probably not as important. There's not that many of them really do get killed by white sharks, and all of them still have to feed. And then all your physiological stuff, they ask the question, gee, what's most important in the survivorship, the ecology, the behavioral ecology, and movements of any of these three species? And it seems to me that physiology rocks, and it's way more important than prey. The prey are way more important than predators.
I think that one thing I've learned in Bar's lab from Bar physiology is the importance of physiology and all this stuff. And it's something that I think really is fundamental. And what we're trying to move towards is, we can build these habitat models based on tagging, habitat association, which are based on your data. We want to come up with this kind of mechanistic model which says, okay, there's certain models where they can work under, and they can or can't be there. And then you can compare them, say, and then you can ideally have forage availability, you can compare and contrast, and what is actually driving this pattern. With some of the recent work in our lab, they built this energetic landscape of tuna, where they were looking at estimating how much they're eating from all the wild tracks and see where they're being developed and how they're driving movements and all sorts of things. I think when you put all that together, it's going to be a constant, cost-benefit analysis. How expensive is it to be there versus how much you're getting.
But you can measure the physiology, you can measure the sea surface temperature, you can measure temperature down deep, you can even measure productivity, but you can't really estimate very well prey availability as a metric. And predation by white sharks or any other predator in any kind of quantitative way. So you've got more information that drives this model that's physiological and water chemistry temperature. than you ever will have for ecological things like predators and prey.
Well, hopefully we're, effort is being put into trying to generate habitat zones for prey. That's always what's missing. If you can build those models, then you can estimate where the prey are. But that's hard to get. And that's people are trying to do Mako sound and where the prey are. Sure. Those things were always like, you have the sharks moving around because they're eating. Okay, what they eat are there actually sardines that eat or whatever. It's amazing how hard it is to find it. But those things are all playing, yeah. Yes, I agree.
Can you generalize it? I'm trying to put it together. If you were to divide the Pacific in half, eastern boundary currents and western boundary currents, does that mean eastern boundary current being like upwelling?
Yep.
Eastern boundary current, big slow sharks. Western boundary current, small fast sharks. Is that? Am I getting all?
Well, no, I mean, so that's it for land and for predators. Not really, because actually all the big pregnant female white sharks I've ever seen are over Japan. And salmon sharks are over there as well. They go to strand. Those are reported sex-free salmon sharks in Western. But yeah, again, it's like the reality is they can be small and in cold temperatures, they're just not stable. But you're not going to get those kind of cold temperatures in Western boundary currents, so you're going to have no limits in terms of where those pops can be from a thermal perspective. Whereas the eastern boundary, you're going to keep that very seasonal.
Well, I was just thinking of the size distribution in general of the shark population as a whole, or the community, because all the species on warm current. There's probably going to be a greater mix of large and small, and I don't know if you consider it selection, but the cold temperature is only is disallowing the small sharks.
No, I think that's totally true. If you say, like, let's look at the California current, you don't see small salmon sharks here hardly ever unless they're dead. You don't see big white sharks, small white sharks up here very often. So I think that's right. I think you have to think of like a sea surface temperature map. There's not going to be very many small sharks where it's very cold. But once that water temperature warms up, those small sharks can probably be dead. So yes, I mean, that's totally beside the thing. And the other question is like, why don't white sharks go up to Alaska where they are? So there's like kind of northern extent for white sharks. But they go all over there and see what they don't stay there. You don't ever see this very sporadic events. And one thought is that that's going to be, if you look at the dissolved oxygen profiles in the subarctic gyre, it's really oxygen drops very quickly out in the Gulf of Alaska. So maybe this combination of low oxygen, high metabolic rate, they body just can't get enough oxygen to fuel their big body size, whereas the salmon sharks are small enough to kind of not have to worry about that. So there's some interesting kind of interactions going on. I don't want to think about why he said it's part of the environment.
I think in the western sort of current and the nurseries and stuff like that, and then these more treated because there's less of these.
Well, I mean, so
yeah,
I mean, I would say the thermal issue is not going to be a problem. But what's actually driving those areas is actually pretty much a subset of all that. It's going to be particular substrate types, it's going to be a range of temperatures, and that's going to be, I think, what defines even the California current. For the white sharks, for example, that was calculated for the California current. No juvenile white sharks don't go offshore pretty much within top 100 years. They're all very short. So realistically, you take that map and cut everything deeper than 100 meters off. That would be a pretty good estimate of habitat for juvenile white sharks. But so like that's an envelope that we're talking about. Within that, they're going to be in a very narrow range of that. But that thermal effect, the limitation is not going to be operating.
Just off the cuff, what do you think is going to happen if climate change does have these impacts on distribution in terms of things like shark attacks on the coast? You got a guy you laughing at this shark attack. I mean, if the white sharks are going to be close to shore where people are, the attacks should go down.
Is there going to be
Or farther north or farther south, depending on.
Yeah, there needs to be more people that have
this will be more of it.
Yeah. There'll be more habitat for people to swim and more habitat for sharks to swim. So, perspective a lot.
Was this year a high year shark test?
What's that?
Wasn't this year an elevated year shark test?
This coast, no. Not this coast. Elevated year.
Elevated was that last.
And has anyone heard about salmon shark stranding?
You're getting seahorses at 1 second. Yeah.